Hi, this is Joshua Gordon. I'm the director of NIMH. And to give you a brief introduction to the NIMH, although it's probably unnecessary for many of you, and to our interests in this area. I'm going to start with an overview of the NIMH, in particular, focus on the challenges in mental health research and treatment that we see broadly, and then talk about opportunities for research on psychedelics, and then introduce the workshop. So let's start with the overview. The NIMH is, of course, the principal federal agency responsible for research on mental illnesses. And our mission is to transform the understanding and treatment of mental illnesses through basic and clinical research, paving the way for prevention, recovery, and cure. In order to accomplish our mission, we support more than 3,000 grants and contracts at universities and other institutions across the country and really around the globe. We also have in our intramural research program about 600 scientists working on the NIH campuses to advance mental health research. And we have a lot to do to advance mental health research and to accomplish that mission. One of the reasons we have a lot to do is because of the tremendous burden of mental illnesses. We're in the top 10 in terms of causes of disability throughout the United States. And indeed, the most expensive illness in the world from the perspective of disability is major depressive disorder, which is the #1 cause of disability globally. Our disorders are so burdensome because they strike the young and because they are chronic illnesses, typically recurring throughout a lifetime, and thus creating a tremendous amount of disability. Now, we do have treatments for these disorders that can lessen that burden, but we have to acknowledge that these treatments are, at best, only modestly effective. About 50 to 60% of individuals with any given psychiatric disorder will remit with the treatment that we have to offer. And in fact, in some disorders, such as depression, with the most aggressive of treatments, we can achieve almost 90% remission. However, even when individuals do remit, the likelihood that they will stay well is unfortunately not great. This study of individuals with depression treated with 4 different antidepressants shows that regardless of what happens over the next year, at most about 50% will stay well over the course of that year following remission on treatment. So we have tremendous burden and our treatments are modestly effective. And of course, the other challenge we have is in particular the worst outcome of mental illness, death by suicide. And what we can see is that over the past 20 years, suicide rates in the United States have been rising fairly consistently, with the potential for a dip starting in 2019 and again echoed in 2020. All this means that we need to do better in terms of how we can treat individuals with mental illnesses, and in particular, individuals with depression, post-traumatic stress disorder, and other disorders that, from the early data, may indeed be responsive to psychedelics. Now, what are the opportunities for research on psychedelics? Well, NIMH has been supporting research on psychedelics for some time. And indeed, the first psychedelic to result in any kind of FDA approval for medication derives from ketamine. We've been supporting research on ketamine both at the NIMH in the intramural research program and at extramural sites for a couple of decades. And in fact, it does seem like ketamine can be an effective therapy, rapidly acting therapy for treatment-resistant depression. Not only ketamine, but of course, the S enantiomer of ketamine, S-ketamine, which was developed by a drug company and tested and shown to be effective in treatment-resistant depression through a randomized clinical trial and is now FDA approved. One of the major advantages of ketamine and potentially of other psychedelic medications is the fact that it seems that they work more rapidly than most antidepressants. And that might be especially valuable if we want to try to prevent suicide. As you know, a significant clinical problem is that individuals come in with severe suicide risk, typically expressing suicidal ideation. And other than placing those individuals in the hospital, we have very little that will reduce that risk in the immediate term. Thus, we've set out to support a range of different studies aimed at using fast-acting antidepressants to try to reduce suicidality acutely in the immediate emergency settings that both youth and adults are seen in with suicidality. 6 of these studies use intravenous or intranasal ketamine or ketamine derivatives in order to test whether that will reduce acute suicidality acutely and whether they can be used in real-world settings. That's one example of a pathway for use of psychedelics in NIMH-funded research. Another pathway, of course, is in understanding the neurobiologic effects of psychedelic medications, including ketamine, but also including others. For example, this paper published in Neuron shows that psilocybin has rapid and persistent effects on the growth of dendritic spines in prefrontal cortex in vivo in mice. And the effect of psilocybin is to enhance the growth of these dendritic spines in vivo. Similarly, another study of DOI, another 2A agonist that is active at the same receptors that psilocybin and other psychedelics bind, also enhanced the formation of spines and increased plasticity in the brain, both measured behaviorally through an extinction paradigm and physiologically through a long-term potentiation pathway. So we can think of NIMH's interests in studying psychedelics, both in terms of proving that they work and also in terms of demonstrating the mechanism by which they work. And in fact, the mechanism focus is really part and parcel of the drug development priorities throughout NIMH when we think of how we want to try to study novel therapies. So NIMH has a range of different funding opportunity announcements and other expressions of our priorities aimed at a mechanistic focus, a mechanistic approach to drug development. These announcements start with genetic discovery and run through understanding of those genetic discoveries through convergent approaches in neuroscience, to drug screening programs that are aimed at targets identified through genetics and other biological pathways, chemical synthesis programs, a network for development of small molecules into clinically useful first-in-human drugs, assay development, and then early stage testing of pharmacologic interventions for the treatment of mental disorders. We also sponsor first-in-human and early-stage clinical trials, and through our small business network, try to help companies that are interested in developing these drugs. All of these pathways can and have been used to support psychedelic research in mental illness research. But we have to acknowledge that the NIMH has not played a significant role to this point in the study of the more recent developments with psilocybin and MDMA and other drugs as they are being taken up by nonprofits and other companies to try to demonstrate their efficacy in disorders such as PTSD and depression. And that's why we have convened this workshop. This workshop, aimed at identifying gaps, challenges, and opportunities in the research areas around psychedelics and mental illness, was convened actually at the behest of a number of different stakeholders, including individuals from the extramural community who urged us to do this, individuals from the advocacy community that would like to see psychedelics developed if they are effective as therapeutics, and individuals from the Congress who have urged us to try to understand what could be NIMH's role and NIH's role more broadly in developing psychedelics as therapeutics. The workshop has three themes. First, we want to understand the roles of basic and translational research in identifying and examining the mechanisms by which these agents may exert their potentially therapeutic effects. Second theme, clinical trials, will examine the clinical data up to now to try to get a handle of what we know and what we don't yet know about whether these drugs work, for what disorders they might work, and how they might be or must be applied in order to achieve therapeutic efficacy. We will consider also how these clinical trials were conducted and how that impacts the data quality, the potential biases involved, and the ultimate conclusions we can draw from those clinical trials. The third theme would be to understand how we might recommend that future clinical trial design be optimized. We want to consider how clinical trials can meet the FDA standards for demonstration of efficacy despite the serious confounds encountered when studying these drugs. And if the efficacy is demonstrated, how we might be able to study how to implement these treatments in a manner that will have the greatest impact on the largest number of people. We will examine the mindset of participants and the environment and settings under which they are exposed to potentially therapeutic psychedelics. And we will discuss what guidelines might be needed to reduce risks involved in using these drugs if they were to enter into widespread clinical practice in the future. So I'm really looking forward to the outcome of this workshop, to the discussions we will have, and to hearing the latest data from all those who are here conducting the trials aimed at trying to understand how therapeutic these drugs can be and how they might be used. With that, I'll turn it back over to the organizers and look forward to the next couple of days.
Thank you very much, Josh, for setting the stage for these next 2 days, and welcome everyone. My name is Steve Zaltzman from the National Institute of Mental Health, and on behalf of my colleagues on the organizing committee, I wish to echo Dr. Gordon's welcome to all of you who've chosen to join us for what we anticipate will be an interesting and thought-provoking workshop on the gaps, challenges, and opportunities presented by research on psychedelics as potential therapeutics for serious mental illnesses and alcohol and substance use disorders. I would be remiss if I did not introduce my colleagues on the planning committee, as well as our stellar logistical team from the Bazell Group, led by Sansere Bowling, without whose magical powers we never would have gotten to this point, and whose continuing efforts over the next 2 days with Kayla Baker's technical wizardry will assure the smooth running of the meeting. I won't repeat Dr. Gordon's description of this meeting's genesis or intended purpose, other than to note that it is our hope and expectation that the presentations and discussions that we'll hear over the next couple of days will provide useful guidance both to the research field and to the National Institutes of Health as to the most productive paths forward. We've been extremely pleased and frankly a bit surprised at the overwhelming interest in this meeting. As of this morning, there have been nearly 4,000 registrants who, as with our speakers, hail from a span of 21 time zones across the globe. This somewhat unexpected but highly gratifying response has caused us to adopt certain logistical procedures to minimize any technical glitches that would interfere with the meeting's accessibility. All of the lectures that you're going to see have been pre-recorded. By contrast, the discussion sessions after each theme will be live, which partially accounts for the timing of the sessions, giving the global dispersion of our lecturers. The meeting will be recorded, and the pre-recorded lectures, as well as the live discussion sections, will be available for viewing in the coming weeks at the website indicated on one of the PowerPoint slides being displayed. Because of the large number of registrants, we are unfortunately unable to guarantee that questions entered in the Q&A box will be answered directly. However, we're confident that our panelists and discussants will provide a wide-ranging discussion after each of the sessions. Now it's my great honor and pleasure to introduce this morning's keynote speaker, Dr. Franz Vollenweider of the University of Zurich. Dr. Vollenweider will provide a brief historical perspective on the advent of psychedelics and then focus primarily on the neurobiology of psychedelic drug effects on self-processing, affect, and social cognition, and the implications of these actions for potential therapeutic applications of these agents. So, without further ado or delay, Dr. Vollenweider.
Good morning. I welcome you to my talk. My name is Franz Vollenweider. I'm a psychiatrist and neuroscientist at the University Hospital of Zurich in Switzerland. Before I start with my talk, I wish to thank the organizer, and particularly Aidan Hampson, for putting together this extraordinary and timely workshop. I would like to give you here a short overview about the history of psychedelic research in Switzerland. Our institute is well known for conducting the first study with LSD soon after its discovery by Albert Hofmann. Initially, LSD was used as a model of psychosis, But soon after, Kondraw conducted the first study using LSD as an adjunct in the treatment of depression. In the mid-60s, two therapy models, the psycholytic therapy using medium doses repeatedly in combination with psychodynamic-oriented psychotherapy, was the model that was mostly used in our institute as well as in Europe. The other model, the psychedelic model, using one or a few high doses that were thought to induce an overwhelming experience that is transformative, was mostly used in the US. In the mid-70s, we had a program into the psychology of psychedelics. Dietrich was interested in the common denominator of drug and non-drug induced altered states, and subsequently, based on this study, he has developed the 5-dimensional altered state of consciousness questionnaire, which in its revised form is widely used in current psychedelic studies. In 1990, I had conducted the first neuroimaging study comparing the effect of psilocybin versus ketamine in healthy human volunteers. Since then, we have conducted more than 50 studies into the neurobiology of psychedelics. The clinical revival using psychedelics started with a study using psilocybin in cancer patients by Charlie Grob and using LSD by Peter Gasser in Switzerland. Both studies gave initial evidence that LSD and psilocybin have some anxiolytic and antidepressant potential. Classical psychedelics can be defined by their primary mode of action. Also, psilocybin, DMT, and LSD interact with various serotonin receptor systems, and LSD also activates the dopamine D2 and D1 receptor. We know today now that psychedelics produce their psychological effects primarily through serotonin 2A receptor activation. In fact, we could block both the effect of psilocybin and LSD with the serotonin 2A receptor antagonist ketanserin in a dose-dependent manner. However, there's also evidence that manipulation of the serotonin 5-HT1A receptor with agonists and antagonists can modulate the effect of psilocybin and DMT in humans. Moreover, we have shown that psilocybin moderately increases striatal dopamine release, a fact or a finding that was correlating with some of the euphoria induced by psilocybin. More recently, using MRS technology, it has been demonstrated that psilocybin increases GABA concentration in the medial prefrontal cortex, as well glutamate concentration in the hippocampus. In fact, a number of animal studies had previously demonstrated that psychedelics increase glutamate release in prefrontal cortical areas that appear to mediate psychedelic-induced neuroplasticity through AMPA receptor activation and the increase of BDNF. In humans, so far, only the increase of BDNF by higher doses of LSD has been demonstrated so far. The psychedelic experience can be characterized and measured along 5 basic dimensions of altered states of consciousness. Using the 5D AAC scale, it can also be further characterized using the 11-DAC scale. At the core of the psychedelic experience lies the phenomena of self-ego dissolution, which comes in two different forms. At medium doses, we most often observe a positive self-ego dissolution, also termed oceanic self-boundlessness. At higher doses, the incidence for a negative self-ego dissolution, also termed dread of ego dissolution, is increased. This ego self-dissolution is accompanied by altered perception, capturing elementary and complex imagery, synesthesia, changed meaning of percepts, and recollection of memories. Two other scales, altered vigilance and acoustic alterations, contribute to the overall effects of psychedelic experiences. Now, the positive self-ego dissolution can be further characterized by different second-order scale scores, capturing loosening of self-boundaries experienced as unity and oneness, disembodiment, positive emotions and bliss, altered space-time, insightfulness, and spiritual experience. It's of note that this positive ego dissolution is also often assessed using the mystical experience scale, which highly correlates with the oceanic self-boundlessness scale. More recently, a more specific scale for assessing ego dissolution and ego inflation have been developed. The spider plot here shall illustrate the dose-response relationship for psilocybin in healthy subjects. Increasing the dose of psilocybin resulted in most of the scales in increased responses on the respective subscales. The 11-DAC also allows nicely to discriminate psilocybin from related drugs such as ketamine. Also, psilocybin and ketamine produce similar overall effects on the 5-DAC. The 11-DAC nicely discriminated the effect of psilocybin and ketamine. For instance, here for the disembodiment, ketamine produced much higher disembodiment than psilocybin. Psychedelics also produce a robust and marked increase in positive emotions. Interestingly, this increase in positive emotions was strongly dependent on the activation of the 2A receptor, as ketamine completely blocked the increase. What I want to show here is that over time, from the first to the 6th hour and the next day, the emotion activation induced by psilocybin, here indexed as emotional excitation or sensitivity, varies across time. The present graph shall illustrate the temporal dynamics and stages of a typical psilocybin experience. Many authors have emphasized that the psychedelic experience is not a firmly defined state, but rather a dynamic process. In fact, many of the subjects report that they appear to progress through different levels of changes along a continuum of perceptual, emotional, and cognitive arousal to culminate in a state of self-dissolution. Until to date, we do not know how the different underlying neurocognitive mechanisms may contribute to the symptom reductions reported in clinical studies. So far, most of the studies looking at the relationship between psychedelic experiences have looked at the content of the experience and how this alteration may predict persisting changes in symptom reductions. However, according to Leiner, the activation and release of often suppressed emotions and the release of personal relevant memories, together with the dissolution of self-boundaries, interpreted as a regression of the ego and a reduction of cognitive control, were most critical for the lasting changes he saw in therapy-resistant depression. Moreover, he emphasized that the post-treatment integration session also markedly contribute to the lasting alterations in behavior. A few recent studies support the view that alterations in the self-ego contribute to the clinical outcome. First, it has been shown that the positive self-dissolution, indexed as mystical type experience, appears to mediate long-lasting increases in openness and prosocial attitudes in healthy subjects. It has also been shown that the overall score or the intensity of the mystical type experience predicted the symptom improvements in anxiety and depression in cancer patients in two larger controlled studies with psilocybin. More recently, it has been shown that the positive ego dissolution, but not the negative experience ego dissolution, contributed to the therapeutic efficacy in treatment-resistant depression. In our own study with psilocybin, we have recently seen that self-dissolution indexed as oceanic boundlessness and the feelings of unity appear to contribute to the symptom reduction in major depression. Neurocognitive models of depression suggest that depressed patients suffer from an increased self-focus and a decreased environmental focus. This increased self-focus has been associated with a negative emotional bias, for instance, expressed as increased negative self-attribution or increased responses to negative stimuli, for instance, fearful faces. On the other hand, the decreased environmental focus may be expressed in reduced empathy and a decreased social interaction. This model had motivated us to study how psilocybin may modulate emotion processing as well as social interaction. Different neuroimaging studies demonstrated that depressed patients show a negative emotional processing bias. They react to fearful faces versus neutral faces with an increased activation of the amygdala. To investigate whether psychedelics affect or modulate emotion processing, we studied the effect of psilocybin on non-conscious and conscious emotional phase processing. We found in the non-conscious condition that psilocybin reduced the processing of fearful faces significantly compared to neutral and happy faces. Again, in the conscious condition, psilocybin also reduced almost double the processing of fearful faces compared to neutral and happy faces. We have then investigated whether psilocybin reduces the amygdala response to fearful stimuli. In fact, we found that psilocybin reduced the bold signal response significantly, particularly in the right hemisphere, and we further found that this reduction correlates with the acute increase in positive mood. based on previous finding, we have then further investigated whether the response of the amygdala during fearful stimuli processing is modulated by the prefrontal cortex or the visual cortex, including the ventral striatum. We found that the coupling between the amygdala and the visual cortex was increased, but that the coupling between the amygdala and the ventral striatum was decreased. This is an interesting finding because we thought that the ventral striatum is involved in saliency detection during negative emotional phase processing. This finding may underlie the phenomena of decentering. Subjects often report that they have a certain distance to their negative emotions, and decentering is a state that allows subjects to have a broader spectrum of emotions and thought patterns. In another study, Fred Barrett has shown that the decreased amygdala response lasts up to 1 month after treatment with psilocybin. Here you see acutely, and then the reduction and how it comes back after 1 month. Furthermore, he reported that he found an increased prefrontal limbic lobe connectivity, which may reflect what we found slightly increased before the lateral prefrontal cortex and the amygdala in our study, but not significantly. He also reported an increased dorsolateral prefrontal activity, which may contribute to the regulation of the amygdala. So far, only one study has investigated the effect of psilocybin on emotional phase processing in depressed patients. Surprisingly, this study reported that psilocybin produced an increase in both signal responses 1 day after treatment. Further studies, and particularly longitudinal studies, are needed to clarify this disparity. Now I'd like to switch to some recent studies we have conducted to approach the altered self or self-experience in psychedelic states. Neurocognitive approaches to the self have been linked to self-related processing. Numerous studies have demonstrated that cortical midline structures overlapping partially with default mode network are involved in differentiating self versus non-self referential stimuli. These regions include the dorsal and ventral medial prefrontal cortex, the anterior and posterior cingulate cortex, the temporal junction, the precuneus hippocampus, and the insula. Self-referential processing is proposed to be a prerequisite for the development of a concept of one's own self, to have a self-perspective or an ego. However, it's of note that in addition to one's own cognition, also emotions and internal and external stimuli, for instance, of body perception, are examined in regard to self-reference. In the initial study, we found that the positive experience self-dissolution, indexed as oceanic boundlessness, correlated with the decreased serotonin receptor 2A receptor binding in cortical regions. So, oceanic boundlessness was in particular correlated with the 2A receptor occupancy in the dorsomedial and dorsolateral prefrontal cortex, in the dorsal anterior cingulate, the temporal cortex, the angular cortex, and the insula, nicely correlating with a number of other studies into self-referential processing. Recent study have shown, in fact, that psychedelics alter resting state functional connectivity in the default mode network. For example, ayahuasca, psilocybin, and LSD have been shown to decrease the functional connectivity within the default mode network. Other studies have shown that, for instance, psilocybin alters the functional connectivity between the medial prefrontal cortex and the posterior cingulate cortex. Moreover, we have found that this reduction in functional coupling correlated with the intensity of the psilocybin-induced oceanic boundlessness. Moreover, we have seen that this reduced functional coupling between these two regions correlated with increased positive and persistent effects we had obtained at 4 months follow-up. This reduction in functional coupling also correlated directly with the positive effects we had measured at 4 months follow-up. Moreover, it's of note that these changes, these persistent changes, for instance, an increase in appreciation of life or in self-acceptance, were driven not just by the overall amount of oceanic boundlessness, but by specific alterations such as positive emotions and the feelings of unity. These findings suggest that psychedelics may mediate lasting changes through different mechanisms, for instance, emotion regulation versus altered self-regulation. A number of recent neuroimaging studies have also investigated the correlation between ego dissolution and alterations in functional network connectivity. However, these studies have not yet yielded a convergent result so far. The most consistent finding appears to be a reduction in alpha power, particularly in the posterior cingulate cortex, that was correlating with ego dissolution in the case of psilocybin and LSD. To further investigate potential correlates of an altered self, we have investigated the effect of psilocybin and LSD on resting state global brain connectivity. Both drugs yielded very overlapping changes of global brain connectivity. Both LSD and psilocybin increased functional connectivity in sensory cortices, but decreased global brain connectivity in association cortices. Interestingly, these changes followed the drug uptake to the peak. Moreover, we found that these changes correlated with the baseline functional connectivity, suggesting that baseline functional connectivity may provide an interesting predictor of individual drug responses. Moreover, we found that the 2A receptor gene expression maps correlated with the LSD-induced changes in global brain connectivity. Finally, we found that the disconnection of the somatomotor network correlates with the experience of disembodiment, a component of oceanic boundlessness. We have recently proposed a system-based mechanism that may explain, at least in part, the psychedelic-induced changes in global brain activity. Pyramidal cell neurons appears to lay at the interface of integrating corticocortical with corticothalamic information flow. We have seen in previous studies that psilocybin and LSD disrupt sensory gating. The disruption of sensory gating has been associated with a disruption of cortico-striatothalamic cortical loops involved in thalamic filtering of internal and external information to the cortex. In fact, this view is supported by recent findings that LSD, for instance, increases the direct connectivity from the thalamus to the posterior cingulate cortex or to the ventral striatum, or that the functional connectivity between the thalamus and sensory areas is increased during LSD or psilocybin administration. However, a simple alteration of thalamic gating does not explain the fact that we also found that functional connectivity between the thalamus and association cortices is decreased. So we do not know whether psychedelics disrupt sensory gating at the level of the cortex or directly at the level of the thalamus, for instance, through interacting differently with different thalamic nuclei. An alternative hypothesis that has been put forward is that psychedelics may disrupt top-down predictive coding, and that this also leads to a relaxation of cortical control over the thalamus and an increase in sensory information flow. It has been proposed that self-representation strongly depends on the experience of a body self. To study the impact of psychedelics on the bodily self-experience, we have used the predictive coding approach. The predictive coding framework of Bayesian learning posits that the brain continuously updates a hierarchical model of the current state of the world to infer causes of its external and internal sensory inputs. According to this framework, the brain learns to model and update predictions across multiple levels of representations, for instance, body perception, and to minimize surprise. At each hierarchical level, top-down predictions based on current beliefs, these are priors, are subsequently compared and matched with bottom-up lower-level representations. A discrepancy between the predicted and the actual incoming bottom-up information produces a prediction error signal. It is proposed that psychedelics may impair prediction error signal processing, for instance, to relax the precision of high-level priors, which may be linked to an altered body self-experience. To study the effect of psilocybin on prediction error processing, a tactile mismatch paradigm was used. To induce tactile mismatch responses, trains of stimuli were applied and switched randomly between double pulses of high and single pulses of low intensity after a variable number of repetitions. The unpredicted stimulus, the deviant, versus the standard, produced activations in brain regions that are involved in body awareness. Under placebo condition, the ventral and dorsal medial prefrontal cortex, the anterior cingulate, the primary somatosensory cortex, the insula, and the thalamus were activated. In this study, psilocybin produced expected feelings of unity as well as the experience of disembodiment. Psilocybin reduced the bold signal response to unpredicted stimuli versus the standard in brain regions implicated in bodily self-awareness. The strongest reduction of the bold response to unpredicted stimuli was found in the ventral medial prefrontal and in the dorsal medial prefrontal cortex, and to a lesser extent in the thalamus. In the co-registrated EEG, psilocybin reduced the so-called mismatch negativity over frontal electrodes. The reduction in mismatch negativity correlated with the experience of disembodiment and the feelings of unity. Taken together, these results suggest that psilocybin alters the integration of tactile stimuli through a top-down aberrant prediction error signaling. In another study, we have investigated the impact of psilocybin on self-other processing. In this study, we used a self-monitoring task involving speech production with feedback varying in source, self versus other, and in pitch. Subjects got a cue to which they had to respond with vocalization almost in real time, and then they had to rate whether they heard their own voice pitch shifted, another voice, or another voice pitch shifted. The study revealed that psilocybin altered a P300 related self-stimuli encoding that was localized to alterations in the anterior cingulate, in the right insula, and in the right superior temporal gyrus, brain regions that were also implicated in the experience of self and body self in other studies. Moreover, we found that the reduction in the P300 related ERP in the anterior cingulate and in the insula correlated with feelings of unity and changed meaning of percepts. So this study suggests that this paradigm may be useful to study the processing of self versus other longitudinally in, for instance, clinical studies before and after application of psychedelics. I would like now to present you a study which we have conducted to investigate the impact of psilocybin on social cognition. In the study, we used a social exclusion test, the so-called Cyberball test, in combination with functional imaging. The participant meets, before he goes into the scanner, two other people with whom he thinks that they play a ball-tossing game. However, when he's in the computer, the game is controlled by the computer, and after some time, when they had nicely interacted, he gets excluded from ball playing. The exclusion of ball playing normally induces in the placebo condition a so-called social pain signal, an activation in the anterior cingulate. At the behavioral level, we have seen that psilocybin reduced the mean exclusion rating. That means that subjects felt less painful when they have been excluded from ball playing. Similar, in the fMRI, we have seen that psilocybin reduced the ball signal response in the ACC when subjects have been excluded from ball playing. Moreover, this reduction in the Bolt signal response in the ACC correlated with feelings of unity. In another test, in the empathy test, we have seen that psilocybin does not increase cognitive empathy, but it increases emotional empathy, that is, the capacity to feel in other people, versus cognitive empathy, which assesses a cognitive process how other people may feel. So this shows nicely that psilocybin may enhance, for instance, the interaction with other people and open up a new space, for instance, in psychotherapy, how subjects may interact with the therapist. This increased emotional empathy also correlated with the altered meaning of percepts, how people perceive the other or the other world. A similar increase has also been reported with LSD. The present study suggests that psychedelics modulate emotion, social, and self-processing at different levels. The reduction of processing of negative stimuli may in particularly contribute to the normalization of the negative emotional bias reported in depression. The reduction of self-processing may help to reduce rumination seen in depression. Alterations in social processing, such as an increased empathy, may contribute to the improvement of the patient-therapist relationship, but may also help to reduce social withdrawal. Moreover, it's important to note that the resting state functional connectivity seen in our study may provide a valuable marker to predict individual drug responses. This hypothesis or this finding has to be tested in further studies. Moreover, it seems that neurocognitive approaches into the self-processing and emotion regulation may provide a useful functional measures to further address the mechanism of psychedelic drug actions and their contribution to the clinical outcome. Moreover, the potential role of attention and the alteration of various cognitive functions by psychedelics, as well as the interaction of cognitive processing and emotion regulation, remains to be investigated. Now I want to stop my presentation, and I thank you very much for your attention. And I want to take the opportunity to thank my group members in Zurich, as well as my international collaborators that contributed in many ways to the results of this study. Thank you very much.
Thank you very much, Dr. Vollenweider, for setting the stage for our meeting's three major themes: basic and translational research, clinical trials, and optimizing future trial design.